In this video protocol, you will see how to dissociate adult mouse lung tissue using gentleMACS. Following dissociation of lung tissue in the presence of collagenase D and DNase I, cell suspensions are filtered using a 70 micron cell strainer, centrifuged, and suspended for further applications.
Hi, I'm Melanie Jungblut from Miltenyi Biotech in Germany. Today I'll show you a reproducible and automated procedure for the preparation of a single-cell suspension out of mouse lung tissue by using the gentleMACS dissociator. We use this procedure in our lab to prepare single-cell suspensions for the preparation of leukocytes or endothelial cells. Okay, let's get started.
Before we begin the protocol, a few standard solutions must be prepared and at the ready. First, we will need Hebe's buffer. Second, a collagenase D solution containing 100 mg per milliliter enzyme and Hebe's buffer. Third, a DNase I solution containing 20,000 units per milliliter DNase I. Lastly, two common buffering solutions. A typical PBS buffer at pH 7.2 and a PEB buffer derived from Automax rinsing solution and BSA stock solution. We begin our protocol with a dissected lung derived from a 6 to 7 week old female Baupsy mouse. Make sure that the lung is free of adjoining organs such as the heart and thymus, efferent and afferent blood vessels, trachea, and connective tissues. Rinse the tissue in a petri dish containing PBS to remove erythrocytes. Typically, a mouse lung at this age weighs between 110 and 150 milligrams. If you plan to dissociate lungs from older animals or other mouse strains, weigh the tissue and buffer. A maximum of 450 milligram tissue can be dissociated per one tube. Transfer the lung tissue to a gentleMACS C tube loaded with 4.9 milliliters of Hebe's buffer. Add 100 microliters of collagenase D solution to a final concentration of 2 milligrams per milliliter to the lung tissue and buffer. Now add 10 microliters DNase I solution. For more than 150 milligram tissue, add 20 microliter DNase I solution. Make sure the C tube is tightly closed and place it upside down onto the gentleMACS dissociator. Make sure that the tissue is located between the rotor and the stator. Select the lung program and run it by pressing the Start button. When the program ends, remove the C-tube from the instrument and incubate the sample for 30 minutes in a 37 degrees Celsius incubator. During the incubation, the tube should be rotated using the MaxMix tube rotator or manually turned every 5 minutes. After the incubation, return the C-tube to the gentleMACS dissociator and run the second lung program. While the program is running, prepare a 50 milliliter tube for collecting filtered cells by replacing the cap with a 70 micrometer mesh cell strainer. Once the dissociator program ends, depending on the distribution of sample material in the tube, you may want to centrifuge the tube briefly to concentrate the sample material. The dissociated cells can be removed from the tube by pipetting through the septum-sealed cap of the C-tube using a suitable 1000 microliter pipette tip. The cell suspension is applied to the cell strainer to remove larger particles or aggregates. Once the solution has drained, wash the cell strainer by adding an additional 5 milliliters of room temperature Hebe's buffer. To pellet the filtered cells, centrifuge the 50 milliliter tube at 300 Gs for 10 minutes. Now, aspirate the supernatant and resuspend the cell pellet in your desired volume of room temperature PEB buffer. Do not decant the supernatant. You can now proceed to your downstream application. Dissociation of mouse lung tissue using this procedure will yield a high percentage of viable leukocytes and endothelial cells. The derived single-cell suspensions were stained with CD45PE and CD146FITC or CD31FITC and analyzed by flow cytometry. This plot shows the enrichment of dendritic cells using CD11C microbeads, a mini-MACS separator, and 2 MS columns.
Okay, I've just shown you how to prepare single-cell suspensions out of mouse lung tissue in a standardized way by using the gentleMACS dissociator and C-tubes. This method allows the efficient and gentle tissue dissociation. When doing this procedure, it's important to properly dissect the lung and process the tissue immediately after dissection. The prepared single-cell suspensions can then be used for further experiments, for example the magnetic labeling of the cells and then the separation of leukocytes or endothelial cells. Okay, that's it. Thank you for watching and good luck with your experiments.